this class we will be dealing with Animal Kingdom Part 2. In Part 2 we will be dealing basically with phylum Protista, Protozoa, and basically with Paramecium and the related organisms as such. So we will look at what Protozoa is, different types of protozoans. Then we will have a look at what fission exactly is, what types of fissions we follow, and then we will have a look at the 4 classes of protozoans based on the locomotional organ in the protozoans as such, and we will also look in detail the classification and about paramecium and diseases such as malaria, leishmaniasis, etc. Yeah. So we'll start with the basic, that is phylum Protozoa. Let's have a look at it. So protozoa are unicellular, eukaryotic, heterotrophic organisms. They are either free living or they can be parasites. There are around 65,000 species of protozoans that have been recorded and categorized in different groups. Protozoans lack a cell wall. However, as they are primitive organisms and they are unicellular, they will not be having any organ systems or organs in them. However, they will have cell organelles that perform various tasks that organs or organ systems perform in higher organisms. So, in many protozoa are involved directly or indirectly in many diseases in animals and humans. Example, Plasmodium, Trypanosoma, Trichomonas, etc. Protozoans have many stages in their life. Some of the stages of the life cycle are infectious. The cyst stage in protozoans is dormant, so that is what something makes them survive in this world in different climatic or habitats, climatic conditions and habitats, and they are resistant to environmental stress. Now they have a stage which is known as trophozoite. This trophozoite stage is particularly infectious and causes diseases in organisms. Let's look at general characteristics of the phylum. Habitat. Protozoa are found in aquatic environment mostly. They live in fresh water or oceans. Some are free living as said before, and some are parasitic in plants and animals. Mostly they are aerobic, but some species do exhibit anaerobic characters. These anaerobic characters or protozoa which exhibit anaerobic characters are found in the rumen or gut of the animals mostly. Okay. Some of the species are found even in extreme environments like hot springs. When organisms like protozoa exist in hot springs, they will be protected by a spore or a capsule. We will have a look at what these structures are and how these are made. So some of them form resting cysts to overcome dry environments. Now size and shape in general. The size of protozoa varies greatly from 1 micrometer to large enough and can be seen by naked eye. When we say about large, we can take an example of foraminifera, where it is up to 20 centimeter in size. So it's such a big organism. Now protozoans lack a true cell wall. They don't have a cell wall. So they are flexible and are found in various shapes. Now, but as the cell walls are absent, they do have a plasma membrane, which is similar to a cell wall, but does not have any rigidity. Cells are enclosed in thin plasma membrane. Some of the species have a shell on the outer surface. In some of the protozoan, especially in ciliates, the cell is supported by a pellicle, which may be flexible or rigid and give the organisms definite shape and locomotion. Any doubts till now? Pellicle is something like a membrane itself. It's just like an outer covering of the organism, similar to a plasma membrane, but it will have bodies attached to it, like flagella or other organisms will be, tibia, etc. will be attached to it. Additional layer. So let's have a check. Now cellular structure. They are unicellular, having a eukaryotic cell. So primitive organisms, they are the first of the eukaryotic cells that we have. The metabolic functions are performed by some of the specialized internal structures. They mostly have a membrane bound nucleus in the cell. That is one difference that we will be looking at. The nucleus is not free. In primitive organisms, when we see, generally the nucleus is free bound, so it is scattered in the cytoplasm. But with protozoa, they have a membrane bound nucleus in the cell. The nucleus, though it is inside a cell or membrane bound, it is still diffused, and you will see scattered chromatin here. Such a nucleus which is scattered will always show a lot of spaces in between the nuclear material, right? So that will be a vesicular nucleus. Nucleus with numerous clear areas due to presence of scattered chromatin or nucleus material is called a vesicular nucleus. Okay. Now there will be a central body called the endosome in the nucleus as well. So that what an endosome is, it's basically an intracellular sorting organism. So what it does, it regulates the traffic of proteins between the organelles of the cell, like lysosomes and the Golgi bodies. So when it's a cellular environment or it's an organism, especially in primitive organisms, there will be free flow of matter or materials easily inside the cell. But then again, when it comes to protozoa, they do have certain regulatory mechanisms or regulatory bodies in there. For example, as we said, the endosome, it regulates the protein movement so that there's no wastage of proteins inside the organism. So maximum utilization of resources is what this tells us this organism does. Okay. Transport na matra important illa. So whatever the organism ingests, right? So it will have to make use of it to the best of its abilities. So it's a modification or a mechanism that the organism has, or the organ system that the organism has, to make use of the resource to the maximum. Adana endosome. So this term, epicomplexans, is a term which we'll use in multiple slides in this chapter, because it represents most of the parasitic species of lower vertebrates or invertebrates. Okay, so all that we'll look at this chapter will come under the class epicomplexans. So this is a term, epicomplexans is a term. Under this category is what all the organisms that we are going to discuss today will come in. All parasitic organisms of lower vertebrates and invertebrates belong to this class of organisms. It's a class, okay, epicomplexans. So nuclei of epicomplexans have DNA. They are not RNA bearing organisms, they are DNA bearing organisms, whereas amoeboids lack DNA in their endosome. So there are certain organisms in this protozoa itself which do not have a rigid cell wall. They will be they don't have a particular shape. They will be just like an amoeba. They can change their shape as it is. Such organisms lack DNA in their endosome. Is it clear? Okay. Now, ciliates have micronucleus. So when it comes to ciliates, generally we must have heard each organism has one nuclei. But ciliates, a class under protozoa, DNA ന്യൂക്ലിയസിൻ്റെ അകത്തുള്ളൊരു പാർട്ടാണ് എൻഡോസോം എന്ന് പറയുന്നത്. സോ അതിൻ്റെ ഇൻറ്റേർണൽ സ്ട്രക്ചറാണ് എൻഡോസോം. വെൻ വി സ്പ്ലിറ്റ് ഓപ്പൺ ദ ന്യൂക്ലിയസ് ഇസ് വാട്ട് വിൽ സി എൻ എൻഡോസോം ആക്ച്വലി. ഓക്കെ, അപ്പം സിലിയേറ്റ്സ് എന്ന് പറഞ്ഞ സാധനം, ദിസ് ക്ലാസ് ഹാസ് 2 ന്യൂക്ലിയ ഇൻ ഇറ്റ്. ഇറ്റ്സ് എൻ എക്സെപ്ഷൻ. സിലിയേറ്റ്സിൻ്റെ ക്യാരക്ടറിസ്റ്റിക് ആണ്. ഇറ്റ്സ് ക്യാരക്ടറിസ്റ്റിക് ഓഫ് സിലിയേറ്റ്സ് ദാറ്റ് ദേ ഹാവ് 2 ന്യൂക്ലിയ. വൺ ഇസ് എ മൈക്രോന്യൂക്ലിയ and one is a macronucleus. So what a micronucleus does is it provides germline nuclei. But then again, it does not express their gene, which means this micronuclei is basically involved in sexual reproduction of the organism. But then again, the characters of this gene is not expressed in the organism as such. Also, there is a larger nucleus, the macronucleus. This provides RNA for vegetative growth. This will be DNA, this will be RNA when it comes for macronucleus. The macronucleus, which is the largest one, controls the characteristics of the organism as a whole. This will be just a supportive role, but when sexual reproduction comes in, this will be the main factor that controls the entire mechanism. We'll look at it. When we study paramecium, we'll see a type of reproduction in paramecium called conjugation. Conjugation is mainly controlled by micronucleus. We'll have a look at it, okay? So, 2 haploid micronuclei formed, fused to form a diploid micronuclei. From this, a few new micronucleus is formed. The old micronucleus is destroyed. Just leave that as of now, park it as of now. When we look at paramecium, we'll have it in detail. The plasma membrane encloses the cytoplasm and other locomotory projections like flagella, pseudopodia, and cilia. Again, a characteristic of this species is that there is a definite bimarcation of endoplasm, ectoplasm, and endoplasm. The cytoplasm is divided into 2. So when we look at cytoplasm, if this is an organism that we are looking at, and this is the plasma membrane, for example, this is the entire cytoplasm. There will be a demarcation of the cytoplasm as well. This part of the cytoplasm will be called an ectoplasm, and this part will be an endoplasm. So just like an ectoderm and endoderm, we will have an ectoplasm and endoplasm. The difference only is that the ectoplasm will have most of the organelles having in it. So this is a highly dense area, the cytoplasm, which will house all the organelles in it, whereas this will be a loose area of cytoplasm, which will be granular in structure. There will be grains or granules in it, but we won't be able to find much of organelles in it. All the Golgi body, mitochondria, everything will be, or vacuoles will be localized in this particular area. It's a highly packed area. Yeah, is it clear? So this is something that we will see from this uh, class of phylum as it is, that the cytoplasm is being divided into two structures. So yeah. Some of the genera have membranous envelope called pellicle, which gives a definite shape to the cell. In some of the protozoan epibiotic bacteria attach the pellicle by fimbriae. So this term, fimbriae is like hair-like appendages present in the bacterial cell wall similar to flagella, but this helps them to attach their host. So it's just to attach the host that they have structures on their plasma membrane or pellicle, that's it. Yeah? So cytoplasm is differentiated into outer ectoplasm and endoplasm, just like I said. Ectoplasm is transparent, endoplasm contains cell organelles. Yeah? Cool. So general structure when we are looking at, we'll see what a gullet is. Gullet is an opening that it has, just like we have a mouth for our eating of our food. Gullet is an opening that they have for the food to pass through, gullet and mouth. Yeah? And this is the oral groove structure. All three will contribute towards the entry of food into the organism. Yeah? So it has a definite anal pore. This is the pellicle lining. This will be the cilia or the fimbriae will be attached to this structure. And food vacuoles and contractile vacuoles are another characteristic of this phylum. When we look into paramecium, we will find two contractile vacuoles. Paramecium is an exception as such. We will look it in detail. Okay.